Okay, so DNA sequence has some regulatory elements that recruit proteins called transcription factors, and these transcription factors recruit the enzyme called RNA polymerase. And RNA polymerase is what actually copies the DNA template into our RNA, and then the RNA gets further processed into our mature mRNA. So now we've gone from the genome to our working copy. Because this is a fairly dynamic process, just to show you again in a sort of little more detail, because also DNA is double-stranded, but we know we have to access the bases of the double-stranded, but you have to open it up to access the bases. So here we have our regulatory region and then our protein coding sequence. RNA polymerase has been recruited to the start. And then the DNA sequence actually opens up. So the hydrogen bonds holding the bases of DNA together break, allow the RNA polymerase to get in there and read the bases. And then as it moves along the strand, the DNA closes up behind the RNA polymerase, so the double-stranded DNA stays intact at the end of this process. And then you have the RNA strand copy being made. So the DNA isn't, at the end of the day, affected in any way. Starts out double-stranded and ends up intact double-stranded. It just opens up briefly to allow the RNA polymerase to come in and copy the bases into RNA. So to actually take a look at the base pairing that occurs, so here's the DNA template. We have, so like I said, RNA and DNA can hydrogen bond. So here's an A of RNA hydrogen bonding with a T of DNA. So why is that? Why do we have this bonding occurring between an RNA strand and a DNA strand?
Because of the hydroxide group?
I mean, so they're basically the same bases, but why is that a part of this process? Is what I was trying to get at.
Are you trying to take the information from DNA?
Yeah, how else are you going to get the information from the DNA? So if your DNA is a T, you want your RNA molecule to, of course, be complementary at an A. So if these hydrogen bond, we know it's the right RNA base being put in. C and a G hydrogen bond. And then here's a U coming in, hydrogen bonding with the A. That's being added to the growing RNA chain. And you can see this chain is now growing with these hydrogen bonds between DNA and RNA. We know that of course, they don't stay bound. As the RNA polymerase moves along and the DNA needs to close back up, the bonds between the DNA and RNA are broken. The bonds reform between the two DNA strands, and now we have our single strand of RNA being made. Okay, so questions about RNA. So I'm going to give you guys a chance to transcribe some DNA into RNA. So this is the first line of the worksheet that was handed out. And then if you get through that, I also have a strand of RNA to see if you can figure out what the DNA strand was that produced that RNA.